Good morning, all. Thanks for giving me a chance to present in this international CME on immunity. So, as we all know, immunity is a combined or it is a wide, a long subject, and based on the immunity, our lifestyle protection from the infective agent is being possible. So, let's start with the immunity, the key for to the long-term protection. Now, immune is a word which is being derived from the Latin immune meaning exempt or free from burden or duty. Now, immunity it is defined as an ability of an organism to defend himself against the invading organisms like bacteria, virus, fungi, and the cells which are expressing abnormal cell markers, mainly the tumor cell. And immunology is a separate branch which deals with the immune system of the body in which the host identifies itself from the non-self antigens. Why immunity is important for long-term protection or body protection? It helps in fighting against the infections, whether it can be due to the bacteria, virus, or parasites. It neutralizes the harmful substances which are acting as a toxins, recognize and eliminate the abnormal cells, mainly the malignant cells, tumor cells, and it is mainly having a crucial role in overall health of the well-being. Now let's study how this immunity is being working in our body. Broadly, immunity is classified into innate immunity and the acquired immunity. Innate immunity is also called as inborn immunity. This inborn immunity is not required any prior sensitization. It takes place as the we birth. There are certain factors which is protecting by birth. These factors are mainly non-specific due to the genetic makeup of our body, offer general resistance, does not depend upon any vaccination or prior sensitization, it comes automatically. So we can say it is a physiological, automatic. So it also has some racial specific, like the malarial parasite cannot infect South Africans because they have a different makeup of the RBC cells. Now these physiological barriers which are mainly protecting or giving an innate immunity, it is based on that is the mechanical barrier, chemical defense and the natural killer cells. Now this mechanical barrier, these are physiological barriers, mainly the skin, mucous membrane, then tears, saliva, hair cells, all these are acting. In addition, that is urine, all these are protecting the body from getting invaded by the microorganisms and will prevent the body from getting infected. Then the chemical defense, it has been provided mainly by the chemical substances which are being secreted in the saliva, then pH of the skin, then gastric secretion. All these gastric secretion, we know that HCl, it is a bactericidal action in sweat secretions which will be secreted, which provides the pH to the skin and helps in preventing the infection. There are certain enzymes in the saliva and the tear. These enzymes are rich in lysosomes, and because they are rich in lysosome, we all know lysosome has a bactericidal activity. Then natural killer cells. These are non-B and non-T type of cells. In addition to this B and T lymphocytes, there is certain cells which are called as natural killer cells. They act as a first line of defense. Acquired immunity we will study in addition. That is, it is like a develop after the birth. Now, innate immunity, the main cells of this innate immunity, these are the neutrophils, which act as the first line of defense, mast cells, natural killer cells, and macrophages. Macrophages, there are two types of macrophages, that is the wandering macrophages. They are moving in the blood and they are surrounded or going through the blood to the all parts of the body. And there are fixed macrophages. These fixed macrophages are present or lodged in the cell or the tissue or the organ. Like example, that is the Kupffer cells in the liver and the alveolar macrophages, microglial cells of the brain. All these are the that is the fixed macrophages. These macrophages are mononuclear in appearance. And as they are mononuclear, the phagocytic system they obey or they play is mainly called as mononuclear phagocytic system. In addition, in the acquired immunity, the main cells which are protecting the body, this is by the T and B lymphocytes. Now, natural killer cell, as I said, is the third category of the lymphocyte, which is neither B, neither T. They constitute about 15% of the total lymphocyte. Their ability is to kill wide variety of the microorganism and the tumor cells. They provide the first line of defense. They are not mature in the thymus, and unlike B and T lymphocytes, they lack the surface antigen, but they carry the surface molecules, mainly CD2, CD16, CD56, and they are negative to the T cell markers like CD3. This is mainly present on the T lymphocyte. These are the large granular lymphocyte, mainly present in the spleen, lymph node, bone marrow, and antibody coated target cells, and kill them by antibody dependent cell cytotoxicity, so which work against the virus and the tumor cells. The main action by which it kills or the phagocytic mechanism take place, it is by the osmotic lysis process by the interferons and causing an active phagocytosis. They have can recognize the process, that is FC receptors, that allows to the kill by the antibody cell mediated, that is the coated virus, and activating antibody antibody dependent cell cytotoxicity. They differ from the other lymphocyte that it does not require prior sensitization, it does not involve MCH antigen killing complex, that is major histocompatibility complex. It activates tumor cells and attack all those abnormal cells which are being not properly marked. Now, the formation of the T and B lymphocytes. The T lymphocytes, these are the major lymphocytes which are present in the circulation, accounting about 70% of the circulating T lymphocyte. B lymphocyte accounts only 15%, and the natural killer cell, as I have already said, it is 15%. These T and B lymphocytes are formed in the bone marrow from the lymphoblast, where they will be maturing in the different part. T lymphocyte mainly matures in the mainly matures in the uh, that is a process in the thymus, and therefore it is called as T lymphocyte, and they process in the thymus. T lymphocyte will be coming into circulation and will be providing mainly the cellular immunity, whereas B lymphocyte initially it was being said it was being maturing in the bursa of fabricus, which is present in the birds, but in human, in the fetal life in the liver, and after birth or in the late fetal life in the bone marrow and the spleen, and they are mainly producing the plasma cells which are responsible for formation of antibodies and mainly the humoral immunity. There are presence of certain antigens on the surface of the T cell lymphocytes, and these are called as cluster differentiation, which gives a numbering which is called as CD numbers. Now, B cells, two types, as I said, plasma cells and the memory cells. T lymphocyte, these are of three types, that is the killer T cells or cytotoxic T cell with CD markers, that is CD8 marker. Whereas helper T cell, it's the CD, that is cell differentiation marker is 4, and these helper cells are of again different type, like the T helper cells, 1, that is Th1, Th2, Th17, and T regulatory cells, which are also called as the suppression for the, that is by the negative feedback mechanism, and in addition the memory T cells. Now, B cells as well as the T cells, they always produce a memory T cells. These memory T cells are basic for the long term protection of the body in the future, That is what we call as the immunity of the person. And in addition, there is the suppressor T cells. This is mainly the C8 type of T cells which belong to cytotoxic T cell group, and they will be suppressing the further action of the immunity, or it gives a negative feedback mechanism. Now in this diagram you can see he pointed something. You can see this CD formation, that is mainly the markers are being present which is being differentiating it from the Th1, Th2 and the other cell. Whereas on the B cell you can see these markers are mainly for the forming the antibodies. So here in short there is a differentiating point between the T cells and the B cells. We all know how it is being processed. Now helper T cells, as I said, they are of different type and its CD number is CD4. These helper T cells, Th1, it is mainly responsible for the cellular immunity, and these Th1 cells will secrete the interleukin which will be helpful in further forming the clones of the T lymphocyte, and these clones of the T lymphocyte will help in the extensive phagocytic mechanism for the action. Then it also secretes, that is interferon gamma. This gamma interferon is mainly responsible for the phagocytic action by release of the lymphokines and further lysis of the microorganism. Then Th2, it is mainly having a role in the humoral immunity, and this mainly secretes or activate interleukin 4, interleukin 5 and interleukin 10. Humoral immunity, it will get activating the B lymphocyte and will cause the formation of the antibody as per which antigens are being activated. Then this Th17, this Th17 has got a major role in forming the more neutrophils to be recruited at the site of infection, and these neutrophils will be released into the circulation by diabetes and the chemotoxic mechanism for, that is the providing the phagocytic mechanism. And T regulatory cells, they are immunosuppressants, mainly release the cytokine, that is interleukin 10, and will differentiate between the self and the non-self antigen. This T regulatory mechanism also provides the negative feedback mechanism for the cellular immunity. Then cytotoxic T cells, these are the CD8 cells. Sorry. These are the CD8 cells, mainly helps in the cell-mediated cytotoxicity. Cytotoxic T cells, cells are mainly divided into alpha, beta and gamma, delta type on the basis of the polypeptide chains which are being attached to the T cell receptors. They kill mainly the virus infected and the tumor cells, induce apoptosis. We all know apoptosis is the self or programmed cell death. Then it is important in the transplant rejection. We have seen many times the skin grafts or the kidney transplant, all this may show the rejection, and this rejection is basically based on the cytotoxic T cells. So what we are doing, giving the immunosuppressant to suppress the activity of the cytotoxic T cells. The memory cells, it preserves the immunological memory and hastens the subsequent immune reaction whenever there is a second exposure. One more important complex or the protein which is been present in the immunity, it is the major histocompatibility complex, or it is also called as MHC protein. These MHC proteins are mainly located on the gene, mainly the short arm of chromosome C. This gene code for the specific MHC proteins. These MHC proteins are glycoprotein in nature and they have a unique characteristic. Because of their unique characteristics, it can identify different type of organism and accordingly it will process the immunity. Now, this mainly helps in functioning of the T cell recognizing the foreign invader. The T cell lymphocyte failed to recognize the antigen in the isolation, so the antigen has got closely associated with MHC molecules which in order to recognize the T cell, and this phenomena of recognition by the T cell with the help of MHC antigen, it is called as the associated recognition. Now, MSC protein was also been referred as human leukocyte associated antigen, or what we call as HLA antigen. So, this HLA antigen is nothing but the MSC protein. Now, there are two types of MSC protein, that is the class 1 MSC protein. It is also called as MHC1 protein, and the other is MSC2 protein. MH1 protein, which is mainly a monomer, is present in all nucleated cells in the body of their cell membrane, and it binds mainly with the endogenous antigen. This MH1 is mainly specific molecule reflection for the graft rejections. So whenever we say that there is a graft rejection, it is mainly by the activation of MH1 protein. MH2 protein, it is heterodimer and is present in antigen presenting cells. These antigen presenting cells are nothing but the macrophages and the dendritic cells. And from the exogenous antigen, it will mainly activate the MH2 protein, and this will activate the cytotoxic T cells and will lead to the cellular immunity. So this cellular immunity is mainly produced by the MH2 antigens. This process is called as the dual recognition or MSC restriction. Now, significance of this HLA antigen and MSC antigen. HLA antigen plays a major role in immunity by recognizing the foreign antigens, histocompatibility antigens in the organ transplantation. Presence of some of HLA antigen is associated with the susceptibility of the certain diseases. HLA antigen typing is used for paternity test. It is a newer upcoming. Now, development of acquired immunity. As I said, acquired. The word acquired means we develop. And as we develop, it is also called as adaptive immunity. This immunity comes after the birth. So, in our due lifelong course, this immunity will be developed. So, we are resistant to certain type of infection and we get developed. So, acquired immunity, it is developed after the exposure. It is antigen specific and memory response is very strong. This acquired immunity is again divided into active and passive immunity. This type of acquired immunity, active means by the active infection or live attenuated vaccines which are being given for development of this immunity. So whenever we give live vaccines, it will produce a small site of infection and will develop the memory cells. Then the acquired immunity, the passive, is mainly via the maternal antibody. We all know that mother protects the fetus by passing of a maternal antibody, mainly the IgG type of antibodies, and in the anti-SIDA. Then the type of acquired immunity that is being passively developed by the injection of the antibodies. These antibodies are developed in some other host cells, and these antibodies are directly injected in the patient, and this provides a immunity to the patient. So it is a passive direct installation of the antibody, that is called as the passive immunity. The acquired immunity is further divided into two types, that is can be produced by the either humoral immunity or by the cell mediated immunity. So you can see that is the gamettate progenitor of cells that is of lymphoid origin present in the bone marrow will either activate the C-mediated immunity leading to cellular immunity, or it will activate the B lymphocyte and will be developing the humoral immunity by providing the plasma cells which will be forming the antibodies and the memory cells, and this is basic how it develops the immunity in the body. Now the cell-mediated immunity, it is the cellular immunity which refers to the specific acquired immunity accompanied by the T lymphocyte, macrophages, and it is effective against the parasite, fungi, virus, cancer cells and the transplanted tissue. There are different steps which are taking place in this cell-mediated immunity. The first is antigen recognition, presentation, and processing. Second is activation and proliferation of the T cells and elimination of invader, that is whatever antigen or a microorganism which is affecting the body. Thus, antigen presenting cells. These antigen presenting cells, they have got some characteristic, like they will be acting mainly this antigen like a first line of defense which attack or which recognize this antigen or microorganism. So these are the dendritic cells like microglial cells, the macrophages, B lymphocyte or B cells presenting antigen with the MSC and the T cell and essential for the that is activation. Now this microorganism or the antigen presenting cells are able to bind to the activated C cells only by the T cell receptor. So this is a antigen presenting cells and this is the T cells, and here this binding will be taking place. So what is this T cell receptor? These T cell receptors are the molecules found on the surface of the T lymphocyte responsible for recognizing the fragments of the antigen that are bound to MHC molecules. It consists of two polypeptide chains, and these two polypeptide chains are being further having an alpha and beta component. These T cells are having almost more than 1 lakh receptor site on its membrane, so they can detect different types of antigens. So 1 lakh receptor site means it have a variety. So it will get activated. Almost 95% of the T cells, the T cell receptor consists of an alpha chain and a beta chains complex with CD3 molecule. Now this CD3 molecule, it is acting as a signal transducer. This T cell expressing these receptors with the CD3 complex is called as AB T cell. So it is a AB T cell is the having that complex. Now CD4 means helper T cell, that is Th1, and CD8 means the cytotoxic T cells. These are associated with T4 and T8 type of T lymphocyte which have an active role in binding with the MSC complex protein with TCR, that is this formed protein by the antigen specific cell will bind and will form the complex. Now, where they are being joining, this portion is called as immunological synapse. So, what is this immunological synapse? It is the being act by the linkage between the antigen MHC protein of an antigen presenting cell on one hand and the CD4 or the CD8 protein of the T cell receptor of the T lymphocyte on the other hand. Synapse means a physiological junction between any two cells. Now this diagram you can see, this is an antigen presenting cell, that is a macrophage. This is the complex that is formed by the antigen MHC protein complex, and this is the helper T cell. You can see this is the CD4 type, and there's two chains, that is the alpha and a beta chain with a cell adhesion molecule. It forms this complex, and this cell adhesion molecule, this together, this is called as the, that is what we call as synapse, that is immunological synapse. Once this complex is being formed, this will be causing activation of the cell type, means the antigen processing and antigen presentation. So you can see this is a macrophage. This antigen is being taken inside the macrophage by the endocytosis. Once it comes by the endocytosis, you can see this will be forming a complex by the lysosomal activity, and MH2 protein which is present inside the cell. This will bind with this vesicle and will form an antigen MHC protein complex. This by the exocytosis forms the receptor on the antigen presenting cell. Once these receptors are being formed, the second steps of will be taking place. You can see these receptors megas are being activated or present on the antigen presenting cell. So this whole is the step 1, that is antigen processing and antigen presentation. Once this antigen presentation takes place, it will be activating the T cell. Now, once this T cell get activated, it will cause a release, a small amount of interleukin 1 will be released, and this will activate the T cell. Now, once this T cell get activated, this will cause more and more release of the interleukin 1, and this interleukin 1 will form, that is the clones of T cells. These clones of T cell is called as activation and proliferation of the T cells. Now once this large number of T cells will be formed, this will go and attach to the microorganism and will cause the release of the or killing of the invader. Now this killing of the invader may take place by the osmolytic activity, that is it will be producing a that is perforins on the cells, and because of the perforins it will be causing lysis, that is the aquaporins will be formed and the extracellular fluid will be invaded and will cause the lysis. Secondly, it will cause release of lymphokines. These lymphokines are the chemical substances which will cause the killing of the microorganism, and thirdly, it will be activating the gamma interferons. This all will cause, that is the lysis of the invader cells, and this together is called as the cell mediated or the cytotoxic immunity. So here you can see this is the complex forming. It activates the 1 T cell, and this activated 1 T cell will be forming a complex substance. So this, in summary, if you see MHC antigen, CD4 cell, memory cell and helper T cell, release of interleukin 2, MHC antigen and will be activating. And finally, there will be insertion of porphyrin in the cell membrane leading to osmolysis, release of lymphotoxin for the tumor necrosis and activation of beta T, that is tissue TNF, beta, release of interferon gamma leading to opsonization and increase in the phagocytosis of the antigen. Humoral immunity. Humoral immunity, as we said, it is being provided by B lymphocyte. Upon activation, differentiation into the plasma cell. Here you can see the Th2 helper T cell is mainly responsible for activation of the B lymphocyte. Now, once this Th2, that is cell helper T cell, will cause release of interleukin 2 and activate the B lymphocytes. Here it will be forming a complex with MH1 and the B cell antigen will be projected. Now once the B cell get activated, it will be forming the giving the signals to the plasma cell for the formation of antibodies, and these antibodies are nothing but the immunoglobulin of different types, and these immunoglobulin mainly are IgG, IgM, IgA, IgE and IgD. In short, we can say IgG, it is main antibodies, that is 75% of the antibodies which are belonging to IgG type. They can cross the placenta and will help in the, that is what we call as active or the passive maternal antibodies which are being providing the immunity to the fetus. It helps in opsonization and complement activation, most abundant central in the secondary response. So there are two responses, primary and secondary, and this IgG is mainly responsible for the secondary response. IgA, it is found in the mucosal secretions, mainly the saliva, tear, milk provide local and secretory immunity. It acts as a first line of defense for the mucosal sites. IgM, largest antibody, it is the pentamer. It produces mainly the response during the primary response. Whenever we give any vaccination, there is a primary response and there is a secondary response. So the primary response is mainly provided by the IgM. It is also a key early responder. ABO antibodies are mainly of IgM type. IgE, it is for allergic reaction and mainly has a critical role in anaphylactic shocks. IgD has got a minor role, that is activation of B cell antibodies. So in general, The different action of the antibody, if we club, it acts as an abstinence followed by phagocytosis of the antigen, neutralization of occupying receptors, precipitation, agglutination, complement activation, and antibody-dependent cell-mediated cytotoxicity involving natural killer cells. Now, lymphokines, cytotoxin, and interleukin, these are the intracellular messengers. Lymphokines, it mainly act as a chemical messenger that is being secreted by the lymphocyte. As I have already said, this will help in lysis of the organism. Monokines, chemical messengers secreted by the monocytes. Cytokines, chemical messengers secreted by the any cells during the phagocytic mechanism. And interleukin, it is a chemical messenger whose chemical composition has been determined. As we know, there are 1 to 12 interleukins. Then function of the several important interleukin, that is interleukin 1 will help in activating the T4 cells, that is the helper T cells. Interleukin 2 act as an endocrine action on the T4 cell, activating them for the proliferation and differentiation. Interleukin 12 promotes cell-mediated reaction, promotes production of the interferon gamma and proliferation of the natural killer cells. Interleukin 4 and 5, it stimulate and transform B cell into the plasma cells. And we know that plasma cells are important for the formation of antibodies. And the secondary response, primary response is a slow response, that is the first when we give the vaccine or the first infection, either it is a live or either it is a dead vaccination or the active infection. So you can see the primary response, there is a short infection and the primary response will reach to the plateau phase and will be again falling, coming back to the that is baseline. This cell activation, that is the B cells and the T cells both will get activated. B cell will form the antibodies, and these antibodies are mainly of IgM type. Now when the same infection or we give a booster doses, this is the secondary response. You can see it will be the, that is the response is very triggered faster and it will go to the peak, and it will, even if it lead to the plateau, it will not come back to the baseline. And therefore in certain vaccination, the booster dose are being mandatory, so they will be reaching and giving, providing the immunity. And this immunity is mainly of IgG type. It is predominant and memory based type of immunity. Now this, ideally the secondary response are mainly, or the how the primary response is being developed is the main basic for our long term protection. Lifestyle and nutritional strategies for the immune supplements. The key recommended for the supporting a healthy immune system, it is balanced diet, rich in fruits, vegetables, whole grain, lean protein, and healthy fats. Then adequate sleep, that is 7 hours for the adult. Regular moderate exercise, stress management technique, either by meditation or yoga. Maintain a healthy weight, that is BMI in a normal range, and limit alcohol, tobacco consumption, practice good hygienic activity. All this will help in boosting our immunity. So in summary, immunity is vital for protecting against disease and maintaining health. Healthy lifestyle choices plays a crucial role in building and maintaining strong long-term immunity. Thank you.
Thank you very much, ma'am, for such an